So, cells. Everything around us, that is, alive at least, is made out of them. But cells have to come from somewhere, so where do they come from? Each other. Cells have to grow, repair, and make copies, and they do that by dividing and replicating. That's how babies are made, scabs heal, or people grow. But before a cell can divide, it first needs to duplicate its chromosomes. So before we get into chromosomes, we need to establish that chromosomes look different for different types of cells. In prokaryotic cells, chromosomes are a single twisted and turned loop, while in eukaryotic cells, chromosomes look like an elongated X. In a eukaryotic chromosome, it is composed of many different components. Chromatin, which is the molecule of continuous DNA, and it coils together with the help of histones, which act as the structural integrity of a chromosome. At the ends of chromosomes, there are telomeres, which protect the ends of the chromosomes from damage, and in the center, there's the centromeric region, which is where the two sister chromatids attach to each other. Every organism has a different amount of chromosomes. Humans have 23. So we're starting with prokaryotes, and they replicate differently than eukaryotes in a process called binary fission. Essentially, the cell's chromosomes duplicate and separate to opposite sides of the cell, which elongates the cell's plasma membrane. Then a cross-wall forms that separates the emerging cells, which divides into two daughter cells. Because only single cell was needed to replicate, it is called asexual reproduction, and the daughter cells are identical to the parent cell. Binary fission is a very fast process, and it can happen around 20 minutes. Just like humans, cells undergo a life cycle called the cell cycle, and we use it to help keep track of cellular growth and division, and it consists of two main phases: interphase and the mitotic phase. During interphase, the metabolic activity is at its highest as it performs its normal functions, grows, and duplicates its DNA. There are three sections within interphase: gap 1, S phase, and gap 2. The gap 1 and gap 2 phases are triggered by growth factors and triggers called the cell cycle control system. The cell will not react unless given a signal. At the end of the gap 1 phase, there is a checkpoint which requires sufficient nutrients, growth factors, an adequate cell size, and undamaged DNA. If the cell fulfills all of these requirements, it will pass on into the S phase. If it doesn't, it will enter a phase called gap 0, which is a permanent non-dividing phase for mature cells, like nerve cells, which don't want to divide. During S phase, the DNA is during the gap 2 phase, enzyme and protein synthesis occurs, lots of ATP is produced, and the centrales replicate. The second checkpoint occurs after S phase and gap 2, called the gap 2 checkpoint. The cell will pass this checkpoint if chromosomal replication was successful and the DNA was not damaged, and that's interphase. So during the mitotic phase, or mitosis, 5 phases occur. Right now there are 46 sister chromatids, or 2N. Starting with prophase, the chromatin previously in the nucleus condenses to form chromosomes that are duplicated. Additionally, the centrosomes, which are made up of centrales, are where the mitotic spindle forms and begins to emerge. In doing so, they also move apart from each other. During prometaphase, the nuclear envelope that held the chromosomes break down and the microtubules begin to attach to the kinetochores of the chromosomes. In doing this, they move the chromosomes to the center of the cell. By metaphase, the mitotic spindle is fully formed and the chromosomes are aligned at the center of a cell in a region called the metaphase plate. The spindles are fully attached to the kinetochores of the sister chromatids as well. Now in anaphase, the centromeres pull apart, expanding the cell. In doing so, the sister chromatids separate. The spindles attached to the chromosomes shorten, while spindles attached to each other lengthen to push the centromeres apart, thus expanding the cell. Telophase and cytokinesis happen concurrently. In telophase, the chromosomes uncoil into chromatin, the mitotic spindle retracts, and the nuclear envelope begins to reform. During cytokinesis, the cytoplasm divides alongside the cell. Cells, however, cannot replicate in a void and must be in contact with some sort of surface to do so. This property is called anchorage dependence and regulates cellular division and growth. Additionally, animal cells also divide off the basis of density-dependent growth inhibition. Density-dependent growth inhibition stops cell division when a cell comes in physical contact with other cells. Hence, its growth inhibition is dependent on the density of cells. When these properties are ignored by cells, they often develop into cancer, dividing and growing without regard to contact or the amount of cells. Additionally, cell division is also influenced by growth factors, which are proteins secreted by certain cells that stimulate cell growth. A particular growth factor that regulates the cell cycle are cyclin-dependent kinases. Kinases are consistently found in the cell. However, they require another protein, cyclin, to be active. Cyclin concentrations vary rising and falling, which corresponds with cellular activity. Together they form a complex called a maturation promoting factor. The kinase has a phosphate which, once conjoined with its target protein, transfers that phosphate and alongside it energy for the protein to go forth and regulate the cell cycle. After this happens, the cyclin is destroyed. When cancer cells evade destruction from the immune system, they become a tumor. If the tumor stays in the region in which it developed, then it's called a benign tumor. But if it develops into neighboring tissues, it's called a malignant tumor. This process is called metastasis and signals blood vessels to supply it with uncontrolled nutrients, killing other cells and creating uncontrolled growth. To prevent this, there's a process called apoptosis, in which signals can induce a cell into programmed death. Apoptosis is also used to get rid of damaged cells beyond repair in adults or eliminate unwanted cells in developing children. Now, during mitosis, somatic cells would replicate. Somatic cells are body cells, while sex cells are called gametes. Meiosis is the process that gametes undergo. The main purpose of meiosis is to reduce a cell from diploid to haploid, or 46 sister chromatids to 23. Each human cell has 23 unique chromosomes. The X and Y chromosomes are called sex chromosomes and determine biological sex. All other chromosomes are called autosomes. Most animals are diploid, or 2N, and their somatic cells have homologous chromosome pairs. However, gametes, which are sperm and egg cells, only have one set of chromosomes and they are called haploid. When two haploid gametes merge in a process called fertilization, they form a fertilized egg called a zygote that develops into a baby. Before we start meiosis, something to know is that a pair of homologous duplicated chromosomes is called a tetrad, which forms during meiosis. On a chromosome, genes are located in specific places called loci. At the beginning of meiosis, there are 92 individual sister chromatids. Starting again at prophase 1, the chromosomes come together to form tetrads. Here, crossing over occurs, which we'll talk about later. The chromosomes coil into their form, the nuclear envelope begins to come apart, and the spindle begins to form. Then at metaphase 1, the mitotic spindles attach to the kinetochores of a sister chromatid, while the tetrads align on the metaphase plate. During anaphase 1, the homologous chromosomes separate from the tetrads while the sister chromatids stay together. They do this because the spindles attached shorten while the spindles connected lengthen. Finally, at telophase 1, the chromosomes move to each pole while the mitotic spindle retracts. Cytokinesis occurs concurrently with telophase 1 and results in two haploid cells with duplicated chromosomes. For the sake of space, I'm only showing one of the cells produced from meiosis, but there are actually two cells going through this process. Meiosis II is very repetitive to Meiosis I. During prophase 2, the mitotic spindle reforms and chromosomes again move to the center of the cell. At metaphase 2, the chromosomes align on the metaphase plate and the spindle is fully formed. At anaphase 2, the centromeres separate the sister chromatids through spindle retraction, and then at telophase 2 in cytokinesis, the spindle fully retracts and the nucleus reforms, and now there are 4 gamete cells with unduplicated. So with chromosomes, there are usually bigger chromosomes and smaller ones. They're unique, and so during meiosis, each gamete will only get one of the type. So one bigger chromosome and one smaller chromosome. That's the law of segregation. With the law of independent assortment, it states that unique chromosomes are inherited randomly compared to other chromosomes. So while you will get one big and one small chromosome, whether it's red or blue won't have an influence on each other. Red and blue in this scenario represent parental and maternal chromosomes. This creates 8.4 million possible chromosome combinations for humans. When tetrads are formed in prophase 1 of Meiosis I, something occurs called crossing over. Crossing over is essentially swapping DNA from maternal and paternal chromosomes. Crossing over helps increase the genetic variability. This only occurs between homologous chromosomes which have the same number. The place in which the chromosomes cross over is called a chiasma, and here the recombination of DNA between homologous chromosomes will occur. When the homologous chromosomes separate after anaphase 2, the sister chromatids are half normal and half recombined. After meiosis I, and the homologous pairs divide themselves into recombinant chromosomes and parental chromosomes. The recombinant chromosomes have a mix of both maternal and paternal DNA, leaning more heavily on one of the two parents. The gametes will get either a recombinant chromosome or a parental chromosome. There are about 1 to 4 crossing over events per chromosome, but there can be even more with larger chromosomes. One last thing before we go. Nondisjunction. Nondisjunction is the failure for chromosomes to separate. This can happen during mitosis, meiosis I, and meiosis II. It occurs when cell cycle checkpoints are ignored and can cause significant side effects. When looking at nondisjunction in meiosis, it can occur in either meiosis I or meiosis II. In meiosis I, nondisjunction inhibits the tetrad from separating. If separation occurs normally in meiosis II, then we'll end up getting all abnormal gametes. While some gametes will gain an extra number of chromosomes from the extra chromosomes from the tetrad, the other half of the gametes will in fact lack a number of chromosomes from the insulated separation in Meiosis I. If Meiosis I goes normally and nondisjunction occurs during Meiosis II, then only half of the gametes will be abnormal, while the other half will be normal. Here, the sister chromatids fail to separate, resulting in an extra chromosome for one gamete, while lacking chromosome for the other gamete produced by the same cell.